So first of all, I want to say how exciting it has been to be here and listen to all of your talks. I feel like it's almost an innovation confessional session. So instead of, mine is also a little bit dry and technical, like you were saying earlier. So instead of digging right into this, I feel like I should confess to you my own innovational pathway. So so my background is in chemical engineering, actually, and I always knew I wanted to be a scientist, but I did chemical engineering kind of as a background. I knew I wanted to get into biology, so I figured chemistry and engineering is a great background. Then I went on to graduate school at Johns Hopkins in the chemical and biomolecular engineering department, but I was actually studying molecular biology of cancer cell metastasis and trying to understand how cancer cells move through different parts of your body to make it, a million miles away from where they started in their own kind of spatial context. And what I ended up discovering was that a lot of the, dogma surrounding how certain molecules work was completely context dependent. And so if you take a cell that you study in a petri dish and you put it in a more, or could say more physiologically relevant, but we don't really know, those molecules that you thought you knew how they were working actually localize differently and function differently. And so I really saw that there was a big gap between the basic research side and the translational research side. So as a postdoc, I went completely in an opposite direction and joined an emergency medicine department. And what I was doing there was really trying to understand what's the problem with measuring the patient. How can we really start to study patients more readily and easily without causing them any harm? But what's wrong with the technology today that we can't automatically get more information out of a patient? So that's kind of what's led me to this, which I'll tell you a little bit about and try and keep it less technical than I had planned. But the idea is really that I want to do systems medicine. I want to attack, clinical problems from both sides, basic understanding and mechanism, but from data that's derived from a patient. And the reason that's important is because patients are kind of their own unique context, right? And as we saw earlier, that their molecular dynamics span, a long time range. So disease is not just a particular instance in time. Disease is a progression. And so we really need to develop technologies that can access patient information without harming the patient, but that can also provide very deep insights into the disease. And this is kind of automatically mandates us to develop technologies that explore patient data space in new dimensions, but also it mandates that we are able to democratize access to these technologies. If we're really going to be able to reach patients, no patient is going to sit around for 10 hours waiting for you to take measurements on them in an emergency room or in a doctor's office. So this is kind of a charge that helps technologies reach underserved populations as well. So my personal passion and the reason this project came about was because, actually it's because of sepsis. So I was in an emergency medicine department, and bacterial bloodstream infections are very common. They kind of affect anybody. If you get a paper cut, you can get a bacterial infection. Depends on your own kind of immune status and that particular bacteria. And within hours, you can become deathly ill. And every hour that you wait, that you don't have an accurate diagnosis, your mortality risk increases. And so this is a situation that kind of poses a very large challenge to molecular technologies. And right now, the current state of the art for detecting bacterial bloodstream infections is still the 100-year-old method of culture. So despite us having, sequencing technologies and molecular detection technologies, it still takes between, it still takes between 4 and 12 days to identify whatever it was that was in your blood. So you can see the timeline. The typical timeline is you're admitted, you have symptoms, of sepsis, so you automatically get your blood taken. That blood goes into a blood bottle and it gets put into an incubator, and it shakes and shakes and shakes and shakes until the bacteria decide to grow. Meanwhile, you're treated with some antibiotic, whatever seems like it might be good for you at the time, based on maybe epidemiological data for the area. Then you may hopefully recover and be discharged within a couple of days. That would be the best case scenario. It's not, it is the most likely scenario, but I think the percentage is like 25 to 50%, depending on where you're at. You can die of sepsis very easily. So by day 4, you might have identified which bacteria was in your blood because it finally decided to grow, and then a technician plated it on different types of media, and one type of media showed a certain color. So you know it's salmonella. And then by day 12, you might know exactly which type of salmonella it was, and that information may be transmitted back to epidemiologists to try and study things like transmission and antibiotic resistance. But obviously, you would have benefited much more if right when you came in, or within a couple of hours of when you came in, you were able to identify what was in your bloodstream, right? So in the context of bacterial bloodstream infections, basically what you need is a very broad-based detection technology like sequencing, where you kind of agnostically are trying to find whatever it is that's in your blood. But it also needs to be specific enough that once you pull that information out, you know very particularly what bacteria it was. It has to be sensitive, because in this case, many people from a 10 ml blood draw only have 1 or 2 bacteria. So you have to be able to pick that up among all of the rest of the DNA that's in the blood sample, that's human DNA. It has to be immune to contamination, because to get the blood, you go through skin, and there's a lot of bacteria on your skin and in the environment, and so there can be contaminating nucleic acids and bacteria that get into the sample. So you have to be able to resolve all of this heterogeneity that is in this sample. So it's a very difficult task, and for a long time people have been working on this from a molecular diagnostic perspective because it would make a big impact. You would also want user-independent identification of whatever that sequence was that you pulled out of the blood, right? Because you don't want somebody having to dig through reams of aligned sequence data and say, okay, I'm 85% sure that it's this, but it could be this because the sequencing lined up this way and such. So these are some of the ideas that we incorporated together and innovated around to come up with a way to do this in a very sensitive manner, in a very fast manner, and eventually portable. We haven't quite made it there yet. But what we did was we targeted a gene in bacteria that all bacteria have, and it just so happens that the areas that we target are conserved between all bacteria, but the gene sequence in between those areas is variable and specific to each individual bacteria. And this is a gene that's used actually to track the evolution of these bacteria. So we can very specifically identify which bacteria it is by those variable sequences, but we can detect all bacteria just by using these, what I'm calling primer sites, to amplify the DNA signal. And then we use a very simple technology to read out the sequence. It only requires heat and a dye. So all we do is we heat up, say there's 3 different sequences of DNA representing 3 different bacteria. We heat up those sequences in the presence of this dye, and because the dye only fluoresces when the DNA is double-stranded, and then it loses fluorescence when the DNA becomes single-stranded as you heat it and melt it, you get these very sequence-specific loss of fluorescence curves, which we call melt curves. And this is what allows us to tell the difference between bacteria 1 versus bacteria 2, because the way that the sequences come apart is a physical thermodynamic difference depending on the sequence. So if sequence 1 is different from sequence 2, they will come apart in a different way as you heat them. And typically, so this melting process has been around for a while, but typically it's done on a population of sequences. What we innovated was we said, because you can have a lot of different types of sequences in one sample, let's separate that sample out across hundreds of different reactions. So we're chopping the sample up, essentially, partitioning it. And by partitioning it to a certain extent, we can guarantee that only a single molecule gets into each individual reaction. And in that way, we can actually, instead of seeing some ensemble average of all of the sequences, we can get down to the level of seeing melt curves from individual sequences. So this is how we're able to resolve, say, if you have a polymicrobial infection, where there's 3 different bacteria in your bloodstream. We can separate those pieces of DNA and individually melt them to identify what the sequence is. And then we integrated this onto a microfluidic chip, and this is where we're at right now. So actually we built our own microfluidic chip, but there are luckily microfluidic companies that are really pushing the envelope in the manufacturability of microfluidics. It used to be that people thought, microfluidics are great, but only grad students can use them because you have to stick bubble gum and duct tape together to get them to work once. We've figured out, or I haven't figured out, but other people have figured out how to really manufacture these things to a high level. And so there's currently a digital PCR chip that's available that has 20,000 reactions. So we're able to take our sample of DNA and split it across 20,000 different wells, and we can actually collect the fluorescence in each of those positive wells and identify the melt curve from those positive wells. So we can see, okay, you have this bacteria and you have 10 of those, and you have this other bacteria and you only have 1 of those. So we're able to quantitatively assess what the profile of the nucleic acids are in the sample. This is, again, still in kind of proof of concept phase, but this chip only costs $15, including the reagents we put in. So it's a pretty exciting idea that we could make this a portable detection technology, and instead of having to rely on a core sequencing machine that's, the size of this desk, and, you have to go into a queue to wait for your sequence to come out, we could actually be sequencing bacteria or what have you, at the patient, really at the point of care. So that's the big idea. The other innovative concept we brought into this was, instead of having to have someone really stare at these curves and say, okay, this looks like it belongs to that bacteria and this looks like it belongs to that bacteria, we automated it with machine learning. So what we do is we can actually generate these training curves. We have E. coli from patient number 1, so we generate training curves of that particular melt, and then anytime that E. coli shows up in another patient, our automated algorithm can recognize it based on its curve. And this also helps us because it eliminates a lot of issues that come up from user-to-user variation. So if somebody in the lab is pipetting differently than another person, or mixing things together differently than another person, this type of approach can overcome small variations like that. So some examples of what we've been able to accomplish so far. This is about to be published, hopefully, but we trained our algorithm on 37 different clinical isolates of bacteria, and we were able to resolve them with 100% accuracy with our algorithm by doing a few tricks here and there. These bacterial sequences were 1,000 base pairs long. If you compare that to typical sequencing reads for deep sequencing, where you can detect the low amounts of stuff, are only about 50 base pairs. So having to align all these 50 base pair increments actually requires you to have a lot of material to start with. Here we're essentially kind of sequencing single molecules at the 1,000 base pair length. For 1,000 base pairs, we can tell a lot of different bacteria apart because of the structure of this gene. And we can see even single nucleotide differences in these melt curves. So some examples of some melt curves that are very similar, but we can automatically tell them apart with our algorithm are shown there on the side. And there's another example. We've also kind of compared this to a couple of other algorithm approaches for classifying data, and we found that our support vector machine learning algorithm does a really good job. And this is across different reaction chemistries, mimicking someone pipetting a little differently or mixing something in a little differently. And this is kind of a proof of concept example of how we can detect mixtures of bacteria down to the single cell level. So this was a mocked sample where we mixed 3 different types of bacteria together. And it turns out that we found out later that our polymerase that we were using in our reaction had carryover DNA. So we actually detected all of these light gray curves as bacterial DNA that we weren't expecting. And then we detected what we were expecting, the purple, blue, and brown curve at the single cell level. And then the red curve shows you what you would see if all of this were all together in one reaction. We also have kind of played around with a couple of other applications. So we're able to detect methylation states of tumor suppressor genes very reliably. We're able to serotype different strains of bacteria for kind of epidemiological purposes. And we're also able to tell apart microRNAs, which are kind of a hot topic in bioresearch. This particular family of microRNAs, you can see the red nucleotides in the table are the only differences between these sequences. And this particular family is known to be very important in inflammation responses. So really, there's no other technology that can do a great job of accurately identifying these sequences because they're so hard to detect these small changes, and also they're so hard to accurately prime. If you're in the molecular detection world, you'll know what I mean, but basically to make copies of these sequences so that you can see them or read out a sequence, it's very hard to accurately make copies of them because some of the changes are on the very ends of the molecules. So our technology can very reliably tell all of these apart. There's 5 replicate curves on each of these colors. So just to show you that there's a variety of applications that this technology could lead us down. So to kind of summarize where we've been going, we really took a very, I guess, unique approach because we innovated in multiple spaces and then integrated to overcome this problem. And we used concepts from microfluidics to machine learning to, thermodynamics of DNA melting to PCR. And we were able to really create a proof of concept for an automated, scalable, rapid, quantitative, and inexpensive sequence identification technique. It's tunable for diverse applications. And again, the future goals are really to allow this to become portable. I'm working right now with some people both in neonatology at UCSD and also in oncology to try and tune this enough so that we could really get some patient information out. One of the interesting application areas for oncology is these drug metabolizing enzymes, which are sensitive to the environment. So if you smoke or eat certain foods, the profile of these enzymes can actually change in real time. And these enzymes metabolize 25% of all the drugs that we have on the market. It's really hard to detect the protein of these enzymes, but it's fairly easy to detect the RNA sequence just before the protein. But the RNA sequence is much more informative than the DNA sequence because for every DNA sequence of a gene, you can actually have, in some cases, 1,000 different RNA sequences from one single gene. So what we're doing with this technology right now is to look at that RNA sequence and understand how these enzymes are changing during the course of chemotherapy for patients. So I think That's about it, except I just have to acknowledge that this was obviously a huge collaborative effort. There's no way that you could pull all of these resources together in these different areas without the help of a lot of people. So a lot of this started at Hopkins when I was a postdoc under, I have a Burroughs Wellcome Fund career award at the Scientific Interface, and I worked in the emergency medicine department. Sam Yang is now here at Stanford, actually, in emergency medicine. And we're still continuing a lot of this work in my lab now at UCSD. Yeah?
I was just curious on what kind of time frame you would have with the melting, like your technique as opposed to the standard technique?
Sure. So our technique involves PCR, which takes about an hour for us because of the microfluidic volumes. The melting process takes about 5 minutes, so the heat and watching the fluorescent curve, and the analysis of the curve also takes about 5 minutes, depending on how good your computing is. So it's within like a 2-hour time frame. Yeah.
That's huge.
Yeah.
With regard to your salmonella example, I feel compelled to point out that were it not for poor diagnosis and treatment of salmonella, you wouldn't be here today, since this institution is actually the largest tribute to infectious disease since Leland Sanford Jr. died of salmonella enterica serotyphi.
Yes, yes.
All right, I have a proposal. Thank you so much.